Hi everyone, I'm Dr. Christopher Jones. I'm one of the lecturers in Chester Medical School. I'm going to be talking to you today about COVID-19. So I'm a virologist. My background is infectious disease, specifically virology. So I thought it'd be interesting for you to have kind of my perspective on COVID-19. I'm not a clinician, again, I'm a virologist. So I thought it'd be interesting to come at the problem from a virology perspective. So what I want to think about is the causative agent itself, so the virus itself, and that's what the talk is going to focus on today. We're going to go through a little bit of virology. Some of it you might be familiar with, but some of it you won't be familiar with. And what I'm going to do is go through that information and apply it to COVID-19. So in the first portion of the lecture, we're going to talk about viruses in a bit of a general sense. So we're going to talk about what actually defines a virus, what we mean by virus. Again, some of you will understand this, but what we're really doing is clarifying some of the key points and making sure that we have this foundational understanding of what COVID-19 is. Because again, COVID-19, the disease, is a viral disease caused by SARS coronavirus 2. So we need to understand what SARS coronavirus 2 is, and to do that we need to understand what viruses are, and we need to understand where SARS coronavirus 2 fits in the big picture. So when we're talking about viruses, we're talking about a huge number of really diverse causative agents of disease that cause a large number of different diseases. And again, these diseases are very diverse. We're talking about a lot of different types of disease. So what we need to do is understand how to kind of make sense of this chaos in a way, make sense of this huge number of causative agents. So again, we're talking about viruses because SARS coronavirus 2, the causative agent of COVID-19, is a virus. So we need to understand what viruses are. I'm sure most of you have done microbiology modules at some point, and you know that viruses are a group of causative agents of disease. They're a group of, we don't really refer to them as microorganisms because microorganisms, we tend to think of cellular organisms as being microorganisms, and viruses aren't cellular, they're acellular. But we group them together with the true microorganisms just because they cause disease in a similar way. So these true microorganisms, whether they're prokaryotic or eukaryotic, so bacteria, the prokaryotes, or eukaryotic microorganisms like fungi and protozoa, these are capable of causing disease by infecting a host, and that's why we call this disease infectious disease, because these microorganisms, or some of these microorganisms anyway, will infect a host, cause disease, and they are then capable potentially of moving from host to host or host to environment to host. And this is why infectious disease is transmissible, so it can be spread throughout a population. If we have disease that is non-infectious, so diabetes and most cancers, these diseases aren't transmissible. They don't spread through a population in the same way. And that's because they're not caused by an infectious agent, and they're not caused by an infectious agent, therefore that can spread from person to person. So viruses are just a group of kind of microorganisms, but not really microorganisms. We tend to refer to them as a group of causative agents, and these are causative agents of infectious disease. It's really important that we understand this group because, and you're going to see this as we progress through the lecture, all of our efforts to inhibit COVID-19, so to inhibit the disease or inhibit the virus itself, so to inhibit SARS coronavirus 2 infection, to inhibit the virus from replicating, are very often centered on our understanding of viruses as a group. So we're essentially targeting the viruses and the diseases they cause. This is what therapeutics are. We're targeting the virus, and we're targeting its ability to cause disease. And when we're talking about bacterial disease, the same is true. So when we're talking about bacterial disease, we need to understand what bacteria are, because when we're intervening on bacterial disease, we're targeting the bacteria and we're targeting the ability of the bacteria to cause disease. And again, that's what a lot of this fundamental information is about in this portion of the lecture. It's about understanding the principles of viruses so that we can then understand our efforts to target these viruses therapeutically, often using drugs, so often using antivirals. And straight away there on the surface, in the most simplest way, it's easy to see why we understand what viruses are, because if we're designing antivirals, we need drugs that target viruses. Antibiotics are inherently different to antivirals because antibiotics are targeting bacteria and bacteria processes, and antivirals are targeting viruses and viral processes. So if we want to design these drugs, we need to understand the causative agents we're targeting. As I said in the earlier slide, one of the fundamental defining characteristics of a virus is it's acellular. So it's not cellular, it's not prokaryotic, it's not eukaryotic, it is acellular. It's not composed of any cells, so it's not unicellular, it's not multicellular, it's acellular. Fundamentally, all a virus is a genome. The genome is sometimes associated with some proteins, often functional proteins. This genome is surrounded by a protein shell. So this is really a structural shell that encapsulates the genome. And this structural shell is sometimes surrounded by a lipid envelope. So a lipid, it's not a cell membrane, it's not quite the same as a membrane, but it's composed of similar things, but it doesn't function as a membrane. It's just an envelope. It just sits there and surrounds the virus particle. So we can straight away see that viruses are quite simple, quite minimalistic. They don't have any of the organelles that cellular life has. For example, there's no Golgi apparatus, there's no nucleus, there's no mitochondria, there's no cell membrane. We just have this very, very simple structure of a genome wrapped in protein, sometimes wrapped in a lipid envelope. And this has consequences for the virus, and it has significant consequences for how we can treat viral infections as well. So the fact that viruses don't have any of these organelles that cellular life has means that viruses are what we call obligate parasites. You've probably heard this term. They are obligate parasites or obligate intracellular parasites. And what this means is the viruses have to infect a cell. So they have to enter into a host cell to replicate themselves. If they don't enter into a host cell, they can't replicate themselves because they don't have the machinery to fulfill any of these functions that are required for life. They don't have the capability to replicate their genome. They don't have the capability to express their genome. They don't have the capability to produce energy. They're insufficient. They're not capable of doing these things unless they are inside a host cell. Fundamentally, the reason is that when they are inside the host cell, they are co-opting the host cell machinery. So they are using the host cell proteins, they are using the host cell enzymes, they are using the host cell resources and machinery. And this is why these very simple agents of disease, viruses, are able to cause quite complex disease. It's why they're able to replicate at all, because they enter into host cells and they fulfill the functions there. And again, this slide is just essentially reiterating what I just said. These viruses are obligate parasites. They are obligated to enter into a host cell and use host cell machinery. And as we'll see in a little bit, this has consequences for how we can treat these viral infections. Something else we have to consider when thinking about therapeutics is how these viruses access the cells they need to infect. So we've said all viruses need to enter into host cells and replicate within host cells because viruses are obligate parasites, and that's absolutely true. So how do viruses gain access to these host cells? We probably know that viruses often infect very, very specific cell types. So then we need to consider about how they gain access to those cell types. So we need to think about infection as being quite a rounded process, and this is the point I'm trying to make in these slides here. We have the common cold here, and the common cold infects epithelial cells of the respiratory tract, so the epithelial cells lining the respiratory tract. The common cold virus needs access to these cells to infect them, and it therefore enters into a new host through the respiratory route, so through the respiratory system. Essentially, it's inhaled, and then once it's inhaled, it accesses the epithelial cells and it is able to infect these cells. It then replicates within these cells. As we've said, it engages in its replication cycle by co-opting host cell machinery. New progeny are then produced, so that once the virus is replicated, new virus particles are produced, and these new virus particles can escape the host and infect a new host. And again, these are being transmitted through the respiratory route, so they're being exhaled, usually in droplets of mucus, and they're traveling through the air, and they're then being inhaled by a new host, so by another individual, and the cycle is going on and on and on. The cycle is continuing. HIV is another clear example of a virus that infects humans. So it infects different cell types to the common cold, of course. So when we're talking about the common cold, as we've just said, the common cold is infecting epithelial cells. HIV, however, is infecting immune cells, and these are immune cells that are circulating within the blood. The virus therefore needs access to these immune cells, and the way it gains access to these immune cells is through wounds or through micro tears in the epithelium that allow the virus to enter into the blood system, where it can access these immune cells directly, so where it can attach to and enter into these CD4 plus immune cells, and it can then replicate, produce copies of itself, produce progeny, and these progeny can then be released into the local environment, and then they are potentially able to find a new host either through direct blood to blood, so again through wounds or breaches in the epithelium, or through the semen, because of course one of the primary modes of transmission for HIV is through the semen. It's sexually transmitted. And the point I'm trying to make in these slides again is that these different viruses are infecting different cell types and are therefore transmitted in different ways. So one of the things we need to understand about coronavirus is exactly which cell types it's infecting, and that will help us to figure out exactly how it's being transmitted from person to person. Now, it probably won't come as a huge surprise to anyone that coronavirus is capable of infecting cells of the respiratory tract. Specifically, it's infecting epithelial cells of the respiratory tract, so it's infecting the cells that line the respiratory tract, essentially. If you remember, when we're talking about our epithelium, we're not just talking about our skin. Our skin is our external epithelium, but our respiratory tract and most of our gastrointestinal tract is also lined by an epithelium. This is just our internal epithelium, sometimes referred to as a mucosal epithelium in parts, because it is mucosal. Therefore, this virus, similar to the common cold, similar to influenza as well, is able to infect the internal epithelial cells, so the cells that line our respiratory system, which is an internal system, so again our internal epithelium. It should also come as no surprise that the route of transmission for coronavirus is respiratory, so it'll transmit from individual to individual on minuscule droplets, so tiny droplets of moisture that are released as an individual coughs or even breathes or just speaks. So obviously when we're breathing and speaking, and certainly when we're coughing, we're releasing droplets of moisture. And if an individual is infected with coronavirus, there's a good chance that there's a significant amount of virus present in these droplets, because again, the virus is infecting the epithelial cells. It's producing large numbers of progeny, so large numbers of virus copies within these cells that are then being released into the local environment, and they can then understandably be transmitted out of the respiratory tract on droplets of moisture. There's also the airborne transmission route. This is a bit, it's not as clear whether it's transmitted by the airborne route. It's potentially transmitted by the airborne route, and the airborne route is slightly different to the mucosal route or the respiratory route, because the virus in the airborne route, it's just free floating, so it's just traveling through the air. It's not encased within a droplet of moisture, but again, the principle is very, very similar. It's being transmitted out of the respiratory tract into the local external environment. And again, obviously this shouldn't be a big surprise. We should know this because the emphasis on wearing masks is inherently tied to this. What you're doing when you're wearing a mask is you're preventing the mucosal droplets, so the droplets of moisture, from being released into the environment. You're also preventing yourself from inhaling droplets of moisture in the environment that other individuals have potentially released. So that's why you've probably heard people say that masks are this kind of twofold protection. They're protecting you if you're wearing a mask, and you're protecting other individuals in the area, because you're preventing the droplets being released from your respiratory tract, and you're also hopefully limiting the droplets that you're uptaking from the local environment. Okay, so just some principles of virology there and a little bit of applying them to coronavirus. It's important now we think about coronavirus in the context of other viruses. So what actually is SARS coronavirus 2 in relation to other viruses? So again, I'm sure we're all up to date on, we watch the news and we have a pretty good idea of what COVID-19 is and things like that. But what I want to do is again approach it from a virology perspective, try and give us a real clear definition and a scientific definition, not kind of a news pop culture definition, more of a objective definition of what SARS coronavirus 2 is. And the next stage to doing that is actually thinking about classification. So we want to think about how different viruses are classified and how SARS coronavirus 2 fits within that classification system. Now, we've touched on virus classification a little bit earlier, and we identified this idea that viruses are acellular. So viruses are a little bit different to the other types of microorganism that you probably studied again in the undergraduate microbiology module. I'm sure you've done at some point. It gets a little bit more complicated than that, because viruses being acellular has consequences, or there's other issues tied in to the fact that viruses are acellular. So as we've said, viruses are acellular. They're nothing like the other microorganisms. They're also nothing like any other form of life, really. Viruses are very, very distinct from all other forms of life, and I'm going to demonstrate that now, and then I'm also going to discuss the consequences of that a little bit. So we have the three domains of life on the right here. What we're essentially looking at is a tree of life. Now, there's nothing at the top of this tree of life where these arrows are pointing to, but what would be here is something called LUCA. So LUCA is the last universal common ancestor from which all forms of life descend. The three domains of life that we have here are the three domains of life that all organisms and microorganisms that we know about can be classified into the archaea, the bacteria, and the eukarya. Within the eukarya domain, you can see some kingdoms that you're probably familiar with. Animalia, right? Animalia kingdom. Plantae, which is plants, so all plants fit into this kingdom. And then fungi, all fungi fit into this kingdom. In the bacteria domain, we have all the bacteria. In the archaea domain, we have all the archaea. So the archaea and bacteria domains are prokaryotic domains. The eukarya domain is the eukaryotic domain. So all eukaryotic forms of life fit into the eukarya domain, whether they're multicellular, like the domains there. Some fungi are unicellular, are single-celled, but most single-celled eukarya will fit outside of these kingdoms, but still in the eukarya domain as protists, so as protozoa and slime molds and things like that. Now again, I'm sure this is just a recap, but the point I'm making here is that all these domains are descended from the last universal common ancestor, so all these domains are related to each other, and that's why we can have a tree of life. That's why the term tree of life exists, because if you map out all these different species, all these different organisms, microorganisms and macroorganisms, they are all related to each other, so they can all fit onto one tree, because they have the last universal common ancestor at the bottom. And that's what phylogenetic classification is. Now, you've probably heard of phylogenetic classification. Phylogenetic classification is essentially classification based upon genetic similarity, and genetic similarity is a consequence or is inherently tied to evolutionary similarity. So the earlier, let's say, two species or two branches of the tree of life separated on an evolutionary timescale, the less genetically similar the end species will be, right? Because they're more diverse, because they spread earlier. That's what phylogenetics is. So if you determine the genetic similarity of different species, you can map their evolutionary origin. And again, you're mapping this into a tree, into a big branching tree that is connected at the base by these three domains, which are themselves connected right at the base by the last universal common ancestor. And obviously I know you recognize these terms on the right: species, genus, family, order, class, phylum, kingdom, and what we've just talked about, domain. These are the terms used for this phylogenetic classification. So when we're using these terms, species, genus, phylum, kingdom, order, domain, class, we're using phylogenetic classification. We're using terms that are determined by how genetically similar different organisms are, and that genetic similarity is also inherently tied to phylogenetics, so the evolutionary similarity of these organisms, because evolutionary pathway and genetic similarity are, they're one and the same thing, right? Because as these organisms diverge, they become less genetically similar. So if you map out genetic similarity, you also have this evolutionary tree of life. So that's what phylogenetics is. There's a problem when it comes to viruses, however, because viruses don't fit on this tree of life. So we said we have those three domains, archaea, bacteria, and eukarya, and we said all organisms fit within these domains. Viruses don't. And that's a significant problem, because this classification system is really, really, really useful, particularly from a therapeutic perspective when we're thinking about these causative agents of disease. So bacteria do fit on the system. Bacteria fit within the domain bacteria, and within that domain we can classify their evolutionary history, we can classify their lineage fairly well, and we can see how all the different bacteria are related to each other. That's really, really useful. It's useful from an academic perspective, so it's just inherently interesting, but it's also useful from a treatment perspective, because we know that bacteria that are similar can potentially be treated in the same way. So it gives us ideas of what antibiotics to use, for example. And let me think of an example of this. A really clear example is TB. So you may have heard of antimycobacterials. So these are antibiotic drugs that specifically work against mycobacteria. One mycobacteria that causes a significant amount of human disease is Mycobacterium tuberculosis, the causative agent of TB. Now, to treat TB, again, we use antimycobacterials. There's another member of the Mycobacteria family. So there's another bacteria that is very closely related to Mycobacterium tuberculosis that also causes significant human disease, however, and this is Mycobacterium leprae, which causes leprosy. Because Mycobacterium leprae is very, very closely related to Mycobacterium tuberculosis, so using this phylogenetic classification, they are grouped very, very closely. It has been inferred, and it is known, it's established, that these drugs that are used to treat TB can often also be used to treat Mycobacterium leprae. And this is despite the fact that TB and leprosy are very, very different diseases on the surface. So the symptoms are different, the site of infection is different, they don't really look much like each other at all. We're talking about Mycobacterium tuberculosis, which is a respiratory infection, and then leprosy, which one of the symptoms is the digits are impacted, the extremities, the peripheral portions of the body are infected because it appears to be affecting the nervous system. So these are two diseases that look very, very different on the surface. However, if we actually look at the genetic similarity of the causative agent, so the bacteria that causes it, we see they are very, very similar to each other. They are both mycobacteria, and therefore antimycobacterials, so this specific group of antibiotics, can be used to treat both infections fairly effectively. And again, the point I was making here is that it's useful for us to classify these microorganisms. It's useful for us to understand how these causative agents of disease are related to each other. And again, there's an issue with viruses there. So the issue is, firstly, that viruses don't fit onto this tree of life. So we don't know how viruses are related to other causative agents of disease. Secondly, the fact that we don't really know how viruses relate to each other. It's very, very difficult to map viruses into one kind of cohesive tree. Even if this tree is separate from the true tree of life, so if this tree isn't the normal tree of life we're talking about, this is a separate virus tree of life, you still can't do it because we can't figure out how the viruses are related to each other. There's strong evidence that viruses are what we call polyphyletic. So that viruses didn't just arise once evolutionary, but that viruses arose multiple times over evolutionary history, which essentially means that a lot of different viruses are not related to each other in any meaningful way. They arose at separate evolutionary time points. They're fundamentally different agents of disease. They're just multiple groups that happen to look the same from our perspective. What we can do phylogenetically is group viruses into specific little families, and we're going to look at that in a second. We can group these viruses into their own little families. However, it's very, very difficult to see how these virus families are related to each other. It's very, very difficult to have a cohesive, comprehensive overview. However, the best we can do is group them into little families of relatedness, and one of those families is the Coronaviridae family. You'll see the suffix viridae used a lot when you're reading up on viruses. The suffix viridae essentially just refers to family. So if we're talking about a virus family, you'll see the suffix is viridae. Pox viridae, the pox virus family. Retroviridae, the retrovirus family, Flaviviridae, the Flavivirus family. So that's what this suffix means. And when we're talking about the coronavirus family, again, suffix viridae, coronaviridae. And again, as we said, we can kind of classify these viruses into families. We can actually do that pretty well. We can group them into little families. Sometimes we can classify these families into orders, so we can group some of these families up. We kind of have 7 groups that we can call orders, so 7 orders that have a whole bunch of families in. However, we don't know how these orders relate to each other, and most virus families don't even fit into these orders, so it's not a very good system. And because we don't know how these orders relate to each other, there's no way for us to use class, phylum, or kingdom or domain. It just doesn't apply to viruses because we don't know that information. So how do we classify viruses instead? So because we can't classify viruses phylogenetically very well, we need to classify them slightly different. So what we do is we classify them based upon their phenotype, so based upon the external features they have. So we're not having this comprehensive classification based upon genetic similarity, based upon evolutionary origin. We just can't do that. So what we do instead is we take the families that we've talked about, so these families that are groups of similar viruses, and we classify these families based upon phenotypic features. So these phenotypic features are outlined here. So these are the phenotypes that we can classify these virus groups based upon. So we can determine the phenotype of the virus family, and we can group them based upon these phenotypes. We'll start off with the second classification here, which is dependent upon the envelope. So we can say, does this virus have an envelope or does it not have an envelope? And this is one type, or this is one kind of aspect of this phenotypic classification: envelope or unenveloped. Some virus families have envelopes, some don't. And this is really, really important because the type of antiviral drugs that will work in inhibiting a viral infection can often depend upon whether or not there is an envelope present. So we know that viruses have to enter into a cell to replicate. Now, viruses that have an envelope enter into the cell in a very, very different way to viruses that do not have an envelope. Viruses that have an envelope fuse with the host cell. This term fuse, we have a process called fusion. Fusion is when the viral envelope fuses with the host cell membrane. Now, an important class of antiviral drugs that you may have heard of are the fusion inhibitors. Now, fusion inhibitors are drugs that disrupt this process of fusion. So fusion inhibitors, understandably, often work against viruses that have envelopes. However, viruses that don't have envelopes can't be treated with fusion inhibitors. Fusion inhibitors will not disrupt the replication of these viruses because they don't partake in this process of fusion. The other classifications we have here are dependent upon the viral genome. So we've said viruses are a genome wrapped in protein, wrapped in a lipid envelope, sometimes wrapped in a lipid envelope, not always. The type of genome present varies significantly. So the genome can be RNA or it can be DNA. So host cells always have double-stranded DNA genomes, right? Not true for the viruses. Viruses can have RNA genome or DNA genome. So some viral families have an RNA genome, some have a DNA genome. This genome can be single-stranded or double-stranded. So the host cell genome, the eukaryotic genome, is double-stranded DNA. Viral genome can be single-stranded or double-stranded and can be RNA or DNA. And this genome can be what we call positive sense or negative sense. Now this applies to the single-stranded genomes. If we think about a double-stranded genome, we have one strand that actually encodes the amino acid sequence of the protein that will eventually be made, right? The protein that is encoded for by the gene. The corresponding strand does not directly encode the amino acid sequence. It's just the complement of the encoding strand. So the encoding strand actually encodes the genes. The corresponding strand is just a template. It just base pairs with the actual encoding strand. And again, the encoding strand determines the order of amino acids in the protein that it's actually encoding. If we have a single-stranded genome in a virus, the single strand can either be the template or it can be the encoding strand. This has very, very, very complicated implications for how the virus replicates. We're not going to go into that now at all. It's beyond the scope. Just the takeaway is that this is one of the ways we can classify the virus, and it's a useful way to classify because it describes aspects of how the virus replicates and also infers what drugs the virus may be susceptible to. When we actually use those phenotypic classifications to group the phylogenetic families, this is what we see. Now, this is quite amazing actually, because what we have here is a comprehensive classification of all viruses. So all viruses will fit onto the two graphics that we have here. So we have the DNA viruses and the RNA viruses, and then we have the phenotypic classification filtering down in a flowchart style, and all viruses will fit onto this classification. And again, this classification is based upon the phenotypic classification that we've discussed in the last few slides. So we're taking phylogenetic classified families, so phylogenetic families, and we're grouping these families based on phenotypic classification. As we've said, the first classification we use is DNA or RNA. So does the virus family have DNA genomes or does it have RNA genomes? So DNA genomes on the left, RNA on the right. Is this genome double-stranded DS or single-stranded? That's the next stage of the flowchart. For the single-stranded RNA viruses, we also classify based upon polarity. So we say, is it a positive sense genome or a negative sense genome? And again, that means, is it an encoding genome or is it a template genome? And the final stage is enveloped versus unenveloped. So do the viruses have enveloped genomes or do they have unenveloped genomes? And once you've reached this level of classification, this is where you place the viral families. And you can see here some very, very well-known viral families that I've put on here kind of to demonstrate these principles. These definitely aren't all the viral families. There's a huge number of viral families. What we have here are just some examples of the different families to show you some examples of each different phenotypic classification. So we have the herpes viridae, herpes viruses. These are DNA viruses that are double-stranded and that are enveloped. Pox viruses are also DNA viruses that are double-stranded and enveloped. So on the far left here, we have herpes viridae and pox viridae. Papilloma viridae, these are DNA viruses that are double-stranded but unenveloped. Again, the human papilloma viruses fit into this group. If we look at the RNA viruses, we have the positive sense, so the encoding strand, single stranded genomes. These, some of them are enveloped, including the flaviviruses and the retroviruses, so flaviviridae and retroviridae. Some of the RNA viruses that are single-stranded and positive sense, so encoding strand, are unenveloped. They don't have an envelope. These include the picornaviridae. And then we have the single-stranded RNA viruses that are negative sense, so have a template genome, and these are pneumaviridae, filoviridae, orthomoxiviridae. So you can see how this classification system works. We use those phenotypic features to classify the phylogenetic families. Now, Coronaviridae, I've said all different viruses fit onto this map here. This includes Coronaviridae, so the coronavirus family fits right here. Coronaviruses are RNA viruses. Their genome is single-stranded. It's a positive sense genome, so their genome is directly encoding, and they are enveloped. So this is the takeaway here. Hopefully it makes sense how this classification works, and this is where coronaviruses fit on this classification. Now, why does this matter? It matters because this gives us indications of how we can treat coronavirus therapeutically. It gives us indications of how we can expect coronavirus to behave. Let's think about DNA viruses and RNA viruses. What's the main difference? Okay, firstly, you can treat them with different drugs. So a lot of antivirals that work against DNA viruses don't work against RNA viruses, and vice versa. And what you'll see is remdesivir, which is one of the drugs that's been used against coronavirus infection, so SARS coronavirus 2 infection, typically works against these RNA viruses. So it's no surprise that we've had some success at using remdesivir to treat coronavirus infections. Secondly, it's important to note that DNA viral genomes tend to mutate at a much slower rate than RNA virus genomes, and there's implications for this, because if we have a virus that can mutate very, very quickly, we have a high likelihood that it can jump from species to species. That might sound familiar. And we also have a high likelihood that it could, or a higher likelihood that it could potentially acquire new mutations when it's in a new host, which obviously concerns us for the coronaviruses, specifically SARS coronavirus 2. So these DNA viruses we tend to think of as being slower, as being older, as having having a lower rate of mutation. Pox viruses, herpes viruses, these are slow old viruses. It's not to say they're not scary and they don't cause significant disease. It's just these fundamental characteristics are slightly different. When we're thinking about the RNA viruses, we're thinking about viruses that cause disease that's just as serious, often more serious, but they also have this high mutation rate, so they're able to acquire additional mutations at a very fast rate, which can be very, very problematic. When we hear about new viruses jumping from host to host, jumping from animal host to human host particularly, it's very, very, very often RNA viruses that are doing this, and one of the reasons is because they are they have this faster mutation rate, so they're able to acquire mutations at a faster rate. So what we want to do now is apply some of this a bit more specifically to SARS coronavirus 2. One way that we can do this is to look at the drugs that have been approved by the FDA, which is the regulating body in the US that oversees these kinds of things. We can look at the drugs that have been approved and think about how they target SARS coronavirus 2. It's kind of a short exercise because there's only been 2 drugs approved for the treatment of SARS coronavirus 2, one of which was approved just a few days ago. Remdesivir, you've probably heard of. There's been a lot of tests, there's been a lot of trials done on remdesivir. Baricitinib is it's used, it's been approved, it's used alongside remdesivir. So it's still interesting, but the main focus is really remdesivir. Baricitinib is used alongside remdesivir. So first off, what is remdesivir? Remdesivir is simply an antiviral drug that specifically targets the replication and genome expression of RNA viruses. So when we look at the viral life cycle, and you remember the viral life cycle is essentially the process that occurs inside a host cell once a virus gains access, we'll see that one of the key events going on is genome expression and genome replication. So to produce progeny, a virus must express its genome to produce protein, and it must replicate its genome so it has genome copies that can be packaged in to new viral particles. So genome expression and genome replication are really essential. A lot of different antiviral drugs will target these processes very specifically. So they will specifically target viral genome replication and viral genome expression. And this is key. The reason they do this is because viral genome expression and viral genome replication often have differences to host genome expression and host genome replication. One problem with viruses entering into host cells and utilizing host cell machinery is that it's hard to target these viruses therapeutically. So it's hard to design antivirals because, again, the viruses are using host machinery. So if you have a drug that inhibits viral processes, often that drug is also going to inhibit host cell processes. And what that means is you're often going to have side effects, you're often going to have toxicity, and obviously side effects and toxicity are a huge issue. They want to be avoided, right? Therefore, it is beneficial to target processes that are unique to the virus or that are fairly unique to the virus or have specific stages that are unique to the virus. So it's quite attractive to target viral genome expression and genome replication because certain elements of this process are fairly specific to viruses, which means you can target the processes with minimal side effects, with minimal toxicity. And this is the viral replication cycle here, and you can see, just as we described, we have the virus entering into the host cell, we have the virus making copies of itself, and then we have these copies being released from the cell into the environment where they can propagate infection. And it's these stages here, genome replication and genome expression, that remdesivir is interrupting. It's essentially an analog. So when nucleic acid strands are being produced, it's genome replication really. However, genome replication is happening, it's fundamentally inherently tied to genome expression. Remdesivir is an analog that's being added onto these chains of ribonucleotides, and when it's added onto the chain of ribonucleotides, so when RNA is being produced and remdesivir is added, it essentially terminates the process. So it disrupts the process, it prevents the RNA from being produced, and that's how remdesivir is inhibiting the genome processing of SARS coronavirus 2. So does it actually work? It does seem to work. There's a lot of contrasting papers, but by and large, the papers seem to mostly agree now that there is some effect. The degree of benefit that's obtained, it differs from paper to paper, which is understandable. I mean, one of the issues is the lack of control groups, and you see this over and over again in these papers. Whenever there's initial results coming out that say remdesivir works, it's very, very difficult to then justify giving a group of individuals with COVID-19 a placebo, so giving them a sugar pill, giving them something with no active ingredient, or with no active component. It's difficult to justify that when you could be giving them remdesivir, when there's, again, some evidence that remdesivir works. So a lot of these studies don't have control groups. However, there's ways around that, and you can do comparisons across studies, and the takeaway is that a lot of these studies seem to suggest that remdesivir is effective to a degree. The other FDA approved drug is baricitinib. Again, this is used in conjunction with remdesivir. It's a fundamentally different drug. So remdesivir is a traditional antiviral drug in that it targets the viral replication cycle, as we just saw. And baricitinib is different because it doesn't directly target the viral replication cycle. It's actually an immunomodulator, so it targets the immune response to the viral infection, so it functions in a different way. Baricitinib is slightly different as well because it hasn't gained full FDA approval. It's gained what we call emergency use authorization. So it's approved to be used in the short term, but it's approved in these specific circumstances. So it's again in the context of emergency use. So one of the things that happens when there's a viral infection is there's widespread immune signaling. So the immune system detects features of the virus that are unique to the virus, so that indicate infection, and it responds accordingly. So pattern recognition receptors, which are immune receptors, detect the virus. These pattern recognition receptors specifically detect pathogen-associated molecular patterns, which are portions of the virus. And once the pattern recognition receptors detect this portion of the virus, they induce downstream signaling events by activating transcription factors like IRF3, IRF7, and NF-kappa B. And IRF3 and IRF7 and NF-kappa B induce the production of signaling molecules, and these signaling molecules signal right through the body, right through often multiple body systems to induce changes, and these changes that are induced are typically the induction of antimicrobial or antiviral genes. They are also to induce inflammation and these other immune responses. Again, for many students, this will be a bit of a recap of immunology modules you've done. If these concepts aren't familiar to you, I would encourage you to learn what PRRs are, what PAMPs are, pathogen-associated molecular patterns, what interferon is. These are really fundamental processes at the core of the immune response, or fundamental elements at the core of the immune response to various infectious diseases. So it can sound on the surface like a good thing. So you could kind of expect me to say, okay, baricitinib is preventing viral infection by inducing these pathways, so by activating these responses. But the truth is actually the flip side. Baricitinib appears to work because it inhibits these processes. So interferon, you can see at the bottom here, interferon is a signaling molecule that specifically activates an antiviral pathway. So it specifically activates a receptor called JAK1, which is of a pathway, JAK pathway, that ends in the production of antiviral genes. And again, okay, great. The problem is interferon does a lot of other things as well. And another problem is that tied to all this is the finding that the activation of these pathways, the production of these interferons is actually associated with detrimental consequences in SARS coronavirus 2 infection. It's on the surface, it's unexpected, but if you delve into the literature around interferon and infectious disease, you'll actually see that a lot of infectious diseases can actually be exacerbated by immune activation in this respect. Inflammation isn't always a good thing. And this probably isn't news, it probably isn't completely new news, because you may be familiar with the idea that a lot of infectious diseases, the pathology they cause is inherently tied to the immune response. So hepatitis B is a fairly passive virus. It doesn't directly cause pathology. It just infects hepatocytes and replicates there. However, the immune response to the virus being present attacks the infected hepatocytes, the infected liver cells. It damages these cells, it kills these cells. It ultimately causes hepatitis, so inflammation of the liver, which can, ultimately down the line lead to cirrhosis, which is liver scarring. So we're fundamentally talking about disease that is indirectly caused by the infectious agent, and the immune system is implicated in that. So it's the immune system that is actually causing the symptoms. Even if we just think about the common cold, one of the symptoms of the common cold is a sore throat. That sore throat is just inflammation of the throat, so it's a localized immune response. So again, the primary symptom, a significant symptom, is not caused by the virus itself, it's caused by the immune response to the virus. And again, the takeaway here is that sometimes the immune response can be detrimental to the host, so it can cause more harm than good, essentially. And it appears like that might be the case in SARS coronavirus 2, at least with respect to interferons and inflammation, because this drug, baricitinib, which inhibits the pathway that interferon activates, so it's counteracting the immune response, and in doing so, it actually appears to be benefiting patients, so it appears to be having a beneficial impact. This is that pathway summarized. So again, if this is a new concept to you, or if it's been, a long time since you've done immunology, just a bit of a simplified version here. We have pattern recognition receptors. They're detecting PAMPs. PAMPs are pathogen-associated molecular patterns. So they are detecting features on the virus, but in this case, we're talking about viruses, but more generally, PRRs can detect PAMPs on bacteria and fungi and all types of microorganisms. The activation of PRRs itself activates transcription factors. These transcription factors induce the production of signaling molecules, and these signaling molecules induce widespread changes in the immune system and also beyond. So a whole range of different cell types can be activated by these signaling molecules. And generally what we're seeing in this activation is inflammation and an antimicrobial phenotype. So the expression of genes that cause resistance to microbial infection. Okay, so thank you everyone for your time. Again, I hope this was useful. I hope it provided you with some background virology that supports your understanding of SARS coronavirus 2. I didn't want to just talk about epidemiological stuff or news updates. I wanted to talk about the virology side of things, because as a virologist, I felt like that was the best thing to do and it would be useful to you. I'm going to upload a list of recommended reading as well, just to support some things I've been talking about, just to also provide you with some further reading, so particularly on these two drugs, remdesivir and baricitinib. If you're interested on reading more about them, you can access the literature I'll put up. Okay, thank you very much, and I hope you enjoyed the module this week. Thanks.